Dear friends, welcome to Biology Made Easy. In the previous video, while discussing the transgenic plants, we have discussed Bt cotton. But in this video, we'll discuss how the golden rice was prepared by the Swiss bioengineer Ingo Patricus. Now this idea came to his mind because most of the people in the developing and underdeveloped countries are suffering from vitamin A deficiency that leads to eye problem. Side by side, most of the people in rural areas, they will suffer from anemic condition. The iron content is also very low. So he thought of making rice, because rice is a staple food. Most of the people take rice. So if these genes can be introduced so that they can alleviate the deficiency of vitamin A and iron content, then the problem will be solved to some extent. So what he did? He took on a rice chromosome. In the rice chromosome, different genes were introduced. I have given the whole chart. You can see it perfectly. For vitamin A, it requires a beta-carotene because beta-carotene is the precursor of vitamin A. So this beta-carotene gene was taken from this daffodil, which is a flowering plant, gives beautiful flowers. So from that, the gene is known as phytone synthetase. In short, it is known as PSY gene. So this gene was introduced. And what is the function of this gene? That will produce beta-carotene, which is the precursor of vitamin A. Now you come to this side. So that will solve the deficiency of vitamin A. But what about to iron? Now you see, the bean plants were taken. In bean plants, there is one ferritin gene. And when this ferritin gene was introduced in the chromosome, it increased the iron content. And when iron content is increased, the anemic condition can be improved to better condition. Similarly, the aspergillus is a fungus. From this, phytase gene was taken and introduced in rice. And what is function? The iron absorption is increased so that the rice plant can absorb more and more iron. Then this is a wild rice. From this wild rice, metalothion gene was taken. And when it was introduced in the chromosome, metalothion protein, its function is it supplies extra sulfur, which helps in uptake of iron. From the soil, it can take more and more iron. That means by introducing these 4 different types of genes to the rice plant, the vitamin A can be produced, the iron content of the rice can be increased, and its absorption uptake from the soil also increased. Now when the plants will produce rice, that type of rice will have all this, both iron and vitamin A will be there. Now you may ask, why it is golden rice? Why that term is given? Because the rice, they were slightly yellow in color. That is why the name golden rice was taken. So this golden rice is very famous because of the 2 things. One is iron, the other thing is vitamin A. After few years, another private company produced again golden rice, but golden rice 2. Golden rice 2. But in this plant, the company didn't introduce the size gene for this beta-carotene. It took one gene CRT1 and taken from one soil bacteria known as Orwina. From this, the gene was taken and that was introduced in the plant. So that is known as golden rice 2. So in this way, the golden rice was produced. Now let us discuss another thing. How the herbicide resistant plants are produced. That is again very important because in the usefulness of the transgenic plants we have given that point. So whenever we grow some crops, it is natural that the weeds come out, and we have to eradicate the weeds either manually or with the help of certain chemicals known as herbicides. So nowadays, herbicides are used to kill the weeds. And one of the important components of these herbicides is glyphosate. This is one of the major components. So when it is applied to the crops, including the weeds, what happens? It affects the EPSP synthetase gene. This is the name of one gene, but you can also call it EPSPS. So this compound, it affects this EPSP synthetase gene in a wide spectrum. In most of the plants it is affected. Even in fungi and bacteria it is also affected. And when this is affected, what happens? The synthesis of aromatic amino acids decreases. And what are the aromatic amino acids? One is phenylalanine, tyrosine, tryptophan. So when there will be shortage of these aromatic acids, naturally the protein content will be affected, the enzyme, everything will be affected. And that is why the plants were killed. But when the weeds were killed, one problem is solved, but the main crop plant is there. To some extent, the glyphosate also affects the EPSP synthetase gene of the main crop plant. So what the scientists did, they introduced this EPSP synthetase gene into the tobacco plant. The first experiment. And by what process? Again through agrobacterium mediated process. They introduced it. So when this EPSP synthetase gene was introduced in this tobacco plant, two things happened. Number one, the weeds were eradicated. Number two, in spite of the effect of this glyphosate, the aromatic amino acid production increased 20 times more than the previous one. That means there is a double benefit. On one side, the weeds were eradicated, on the other hand, the production of these aromatic amino acids increased. So this is again one of the most important achievements in the transgenic plants. Then let us give a third example. The third example is the production of hirudin protein. Now you know that hirudin protein, it is an anticoagulant used in blood clotting. And this hirudin protein is produced from the gene, that is again hirudin gene. This is the gene. And this gene is present in the leech. You know that the leeches are found in ponds, lakes. So when this hirudin gene was introduced into one plant, Brassica napus, and the seeds of this Brassica napus, they accumulated the hirudin protein. So this hirudin protein was extracted from the seeds of Brassica napus and used for a medical purpose. Then another example, plantibodies. Now what do you mean by plantibodies? We know about antibodies, but here plantibodies means the plants will produce antibodies directly. That is why they are known as plantibodies. By introducing different types of genes for cholera or for some common diseases, when those genes are introduced and will take the plant product, directly the antibodies enter into our body. That is why in one of the points I have told you tailor-made plants, where I have given the example of pharmaceuticals. So you see directly they are used here. The plant product is directly used as antibody. That is, they are known as plantibodies. Then another example again I will give you. Protein-rich potato. One of the Indian scientists known as Ashish Datta of Jawaharlal Nehru University, JNU, Ashish Datta, he produced one protein-rich potato, where there is 70% protein. Can you imagine 70% protein? So the name of that potato is AMA1 because this is the gene which was introduced in that potato plant. And where from this gene is taken? It is taken from amaranthus. That means from the amaranthus plant, this gene was taken, and when it introduced in the potato plant, the protein content increased to 70%. So these are some of the examples. Again, there are hundreds of examples, like production of lysine-rich pulse, decaffeinated coffee, then blue roses. Likewise, there are several examples which are produced through this recombinant DNA technology, and these are known as transgenic plants. Then I will give you questions. Now, one of the questions you can write. Number one question you can write: golden rice compensates the deficiency of A choice vitamin A, B choice vitamin B, C choice vitamin C, and D choice vitamin D. Number two, which of the following component is present in herbicides? A choice glyphosate, B choice glycophosphate, C choice glyphosate, D choice all are the major components. So hope you will be able to answer, and you must have understood the things perfectly. And again we'll meet in another video. Have a good day.